Hello students. Today we are going to discuss Unit 11 Shrimp Hatchery Technology from BSc third year, semester 6. The objective of this chapter are to learn the importance of shrimp hatchery technology and their life cycle, to understand the preparation of brood stock and selection of spawners and egg collection, to understand hatching and transportation of nauplii and their rearing, to know about hatchery management, and also to understand the harvest and transport of larvae. So traditionally, shrimp fry are trapped and held in ponds and later collected by shrimp gatherers for stocking in grow-out ponds. With increasing demand of shrimp, supply of wild fry for the increasing number of shrimp farms has become insufficient and inconsistent. So the breakthrough in the completion of the life cycle of commercially important shrimps in captivity, such as the tiger shrimp, the Japanese kuruma, the eastern shrimp, and the banana shrimp, has greatly enhanced mass production of shrimp fry under hatchery conditions. The excellent growth performance of these hatchery bred fry in grow-out ponds strongly shows that the shrimp hatchery can answer the industry needs for ample supply of shrimp fry for farming. From many years of accumulated experience and research findings, the success of a shrimp hatchery depends on all this. One is the choice of a suitable site. First, we need to select the site where we need to establish the hatchery. Then, effectiveness and efficiency of the hatchery design. And the design also should be proper to grow the prawn fry. And then experience of hatchery technicians. Not only we should have suitable site and suitable design, we should also have proper technicians to operate everything. And finally, the efficiency of operational management. So first, if we see the selection of a suitable site for a hatchery, the first one is we need to look into seawater supply also. The seawater used in a hatchery should be clean, clear, and relatively free from silt. The water quality should be stable with minimal fluctuation in salinity. Suitable sites are usually found in sandy and rocky shore ecosystem where there is clean, clear, and good quality seawater all year round. Sites not suitable for hatchery are swamps and muddy shores, where the water becomes turbid during heavy rains or due to turbulence. Avoid river mouths where abrupt lowering of salinity often occurs during heavy rainfall. An added advantage of rocky and sandy shores is that good quality seawater is relatively near the shoreline, thus reducing the cost of piping installation and pumping cost. If it is away from the sea, the piping and pumping it costs so much for us. So the hatchery site should also be free from possible impact from any inland water discharges containing agricultural or industrial waste. If the hatchery is close by agricultural lands, farms and fields and all, from there the agricultural waste and the rainwater, everything that might enter into this hatchery farm, which is not advisable. Then we need to look into availability of spawners. Presence of spawners at the vicinity of the proposed site is of considerable advantage in ensuring consistent supply of spawners, reducing the cost of transportation, which could affect the rate of spawning. Next, availability of power source. Electricity is essential to provide the necessary power to run equipment and other life support system in the hatchery. Although some marine pumps and aerators can be driven directly by handy generators, the shrimp hatchery can therefore be operated in areas without electricity supply also. However, It is more economical to operate it in areas where there is a reliable source of electricity. Installation of an on-site standby generator is absolutely necessary, especially in areas where there are frequent lengthy power failures and fluctuations. Then we need to look into freshwater supply also. Freshwater is essential for daily hatchery operations such as salinity, adjustment, equipment maintenance, and for domestic use. Then accessibility. Ideally, hatchery should be sited in areas where there are active shrimp farming operations so that the shrimp larvae produced can be easily transported and distributed to the growth ponds. Hence, the site chosen for hatchery established must be easily accessible to facilitate communication and transportation. Then, apart from site, labor, then the culture, then the seawater, freshwater, and accessibility, we also need to look into climatic conditions. A hatchery can be established in any climatical condition as long as the required rearing conditions can be adequately provided. So, however, all the commercial hatcheries take full advantage of nature in terms of energy source and supply of good quality water. Sunlight is a basic requirement for hatchery operations, especially in the mass production of natural food used as feeds for the growing shrimp larvae. In the temperate region where adequate sunlight is only available in certain periods of the year, hatchery operations are usually confined to a certain suitable So this is especially so in the case of Soma marine shrimp such as Peneus japonicus and Peneus orientalis, which have a single pronounced spawning season. In such situations, hatchery production is relatively limited to a short period of time. On the other hand, the tropics are endowed with warmer climate and adequate sunlight all year round and have ideal climatic conditions for hatchery operation, especially for many warm water species such as the tiger shrimp, Penis monodon, the white shrimp, Penis indicus, which, which spawn all year around. The optimal temperature for hatchery design may be necessary in areas where there are pronounced long period of rainfalls, which often reduces the intensity of light and they cause turbidity in water supply and lowering of water temperature also. Now, if you look into the hatchery design and construction, production target can be determined based on a market demand and financial input. In the case of species such as Penis monodon, at which the production of fry depends on the availability of spawners from the wild, production target is limited by the supply of spawners. So the design and construction there, we have so many varieties, small scale, large scale like that. So this limits the production of capacity of the hatchery, whereas in Penis japonicus and white shrimps, at which spawners are easily available, production capacity is unlimited. Take capacity up to 2,500 cubic meters can be seen in many large-scale hatcheries in Japan, where Penis japonicus is the primary species grown. However, in most Southeast Asian countries where tiger shrimp forms the target species for hatchery production, tank capacity is considerably reduced due to limited spawner supply from the wild. Hatchery design is aimed at achieving the production target, which determines the size of the hatchery. The tank capacity is based on an approximate ratio between algal culture tank and larval rearing and nursery tanks. Desirable algal tank capacity is 10 to 20% of the larval rearing tank capacity. The capacity of maturation tank depends on the number of spawners needed. Now, size of the hatchery. Generally, the size of a hatchery should be based on its functional requirements, and economic efficiency. So whoever is constructing are planning to have these hatcheries, they should see the requirement and their economic condition also. So based on the level of operation, production output, and financial investment, hatchery practice can be broadly grouped into three categories like small scale, medium scale, and large scale hatcheries. If you look into a small scale hatchery, this is a backyard hatchery, usually owned and managed by the shrimp owner himself, using family members or immediate relatives for additional labor. At home only we can maintain this. The main goal is to supply his own shrimp farm with the required number of fry, and the excess may be sold to neighboring growers also. Usually, the hatchery site is an extension of the farmhouse with floor space ranging from a few square meters up to about 1,000 square meters. In such hatchery operation, the total production capability seldom exceed 5 million post larvae per annum, and the hatchery is operated by not more than two technical personnel. Then large scale hatchery. This type of hatchery is relatively larger than the small scale hatchery. In terms of capital investment, hatchery size, production capability, and scale of operation. While hatchery management is somewhat similar to that of small scale hatchery, the production capability range between 10 to 12 million post larvae per annum and is operated by about three technical personnel and three to four laborers. This type of hatchery is usually put up by small cooperatives to supply the required shrimp fry to its member growers. Private entrepreneurs or government agencies may also establish hatchery of such operational scale to produce fry of sale or distribution to the growers. Then large scale hatchery. This scale of hatchery is commercially run by corporations, national agencies, or cooperative projects. While the capital and operational investment for exceed far exceed that of the medium scale hatchery, production capability of such hatchery usually exceeds 20 million post larvae per annum. Such hatchery is centrally and symmetrically managed and is supported by a pool of not less than 6 technical personnel and 6 to 10 laborers. Then what are the facilities should be there? So hatchery facilities like in designing a hatchery, ample space should be there first of all, provided for the rearing and support facilities needed in the operation. Then a functional hatchery should have the following essential components. What are those? First is maturation tanks. The major constraint in hatchery operation of tiger shrimp is the limited supply of spawners from the wild. Hence, eye stock oblation techniques can be used to augment the scarcity of spawner supply, thus maintaining ablated shrimp in maturation tanks that would ensure a constant supply of gravid females. The shape of maturation tanks can either be circular, rectangular, or oval. The tank capacity may vary from 5 to 40 tons with depth ranging from 1.2 to 2 meters. If the shrimps are kept for less than 5 weeks, bottom substrate is not needed in the tank. The tank is installed with an inlet pipe from the wall and a double cylinder standpipe at the center for drainage. This system facilitates continuous flow through of seawater. Spawning tanks. Spawning tanks should be circular with a flat or conical shaped bottom. Water holding capacity may vary from 50 liters to 1.5 tons. The tank can be made of fiberglass, plexiglass, plastic, or marine plywood. The tanks are used to temporarily hold the gravid females until spawning. This is the picture of 12 tons ferrocement brood stock tank. Then larval rearing tanks. Two types of rearing tanks are being used to rear the newly hatched larvae. In Japan and Taiwan, larger tanks with a capacity of more than 50 tons are being used. In Southern Asia, most of the hatcheries use smaller larval rearing tanks of about 3 tons capacity. Hatchery operators call the latest system of larval rearing as the small tank hatchery system, which originated from the Galveston laboratory in the USA. In the farmer system as the large tank hatchery system. So one is small tank hatchery system and the large tank hatchery system. So this is originated in Japan. So large tank is from Japan and small tank is originated from the USA. Small tank system, it'll have 250 liters capacity, something like that. The larval rearing tank may be circular, rectangular, or oval in shape, with tank capacity ranging from 0.8 to 3 tons. The bottom of the circular tanks may either be flat or conical. Rectangular or oval shaped tanks always have flat bottom. The circular tank is usually 1.8 meter in diameter and 1.2 meter in depth, with a central double cylinder standpipe drainage system, which can be used for continuous flow of seawater. When the larvae reach mysis or post-larval stage, rectangular tank is about 1.5 into 5 into 1 meter in size. The drainage pipe is set at the side of the tank. The drain pipe is also used for harvesting. In all types of tanks, seawater is delivered into the tank through an inlet pipe installed at the top of the tank. Then big tank system. The tanks used are rectangular or square in shape, with capacity varying from 50 to 2000 tons or more, like 5 into 5 into 2 meters or 20 into 50 into 2 meters. The tanks can either be located outdoors or, if located indoors, transparent roofing should be provided to allow for sources of sunlight. In a big tank system, spawning, hatching, and larval rearing operations are done in the same tank. The larvae are reared for 35 to 40 days. Then coming to water storage and filtration tank. The water storage tank is normally elevated to effectively distribute water by gravity to the hatchery. The water storage tank capacity should be at least 20% of the larval rearing tanks. Storage tanks are normally constructed out of reinforced concrete to withstand the water pressure. When the water is turbid, installation of a filtering screen and sand filter until becomes necessary. Filter unit becomes necessary. The filter chamber may be constructed adjoining the holding tank. There are two types of filter systems. Gravity filter, where water is pumped into the filter chamber over the surface of a filter bed and allowed to pass through the filter material by gravity to the holding chamber, which is located under the filter chamber. Then second one is reversing filter, where water is pumped directly to the space under the filter chamber and pumped upward through the filter to the surface and to the holding tank. So in both these systems, the filter chamber usually contains white sand, charcoal, gravel, or all the three as filter material. The advantage of reverse system is that water passes slowly through the filter material and the whole surface area of the filter is utilized. It is easy to backwash by spraying water from the surface of the filter, and the detritus underneath are easily washed out. On the other hand, gravity filter method allows water to pass through the water chamber too fast and do not fully utilize the surface area of the filter unless it is provided with pipe spray water over the whole surface area. The advantage of the gravity filter system is that the filter is easily blocked with detritus after a few days of operation, resulting in turbid water and difficulty in backwashing. So this is the disadvantage because the dust and all everything gets blocked in that. Next is aeration. So aeration is essential during the entire larval rearing process in maintaining sufficient dissolved oxygen concentration in the water, ensuring even water temperature throughout the water column through the turbulence, and also help reduce the ammonia content in the water. Large volume of low pressure air, while an air compressor provides small volume of high pressure air. An air blower runs continuously while a compressor which is equipped with the pressure tank runs whenever the pressure is low. The compressor automatically switches on when the pressure drops below a predetermined level. In a hatchery, low pressure area air available at large volume is more desirable than high pressure air at small volume. Moreover, the hatchery tanks are seldom more than 2 meters deep. Rotary air blowers are not designed for oil-free operation and have a tendency to blow oil particles into the airline, producing oil slicks on the surface of the water. Air filters at the inlet and outlet pipes are therefore needed. When the capacity of an air blower is less than 10 HP, ordinary air filters for automobile may be used at the inlet pipe. However, for blowers with high horsepower ratings, synthetic foam can be used. Adjustable pressure tank with resin glass beads or bagasse as filter material are used for the outlet pipes. The Roots blower is more appropriate for hatchery purposes because it seldom breaks down, less complicated to use, and does not produce oil slicks. In culture tanks with depth less than 2 meters, an air pressure of about 0.2 to 0.3 kg per cubic centimeters and a volume of 4.5 liters per meter square per minute is sufficient to oxidize the dissolved organic matter in the tanks. Since continuous aeration is essential to the survival of larvae in high density, any prolonged power interruption would seriously affect the culture organisms in the tank. Thus, it is essential to install an automatic switch which starts a standby generator whenever there is a power failure. A battery operated warning device to signal the crisis and the required operation of the standby generator can also be used. Next, seawater supply and piping system. Seawater can be drawn directly from the sea or from a sump pit. If the course of the water is relatively clear, the water can be pumped directly into the overhead filter tank and stored in the reservoir or storage tank. Water is then gravity fed to various culture tanks through its delivery pipes. However, if the water is turbid and contains a high concentration of suspended solids, the water is pumped first into a sedimentation tank where the suspended solids are allowed to settle down and the clearer water on the top is pumped into the filter tank. Sometimes, when the water source is far from the shore due to low tide, and if large quantity of water is needed continuously, the intake pipe should be laid horizontally from the littoral zone through the underground pump pit and the filter tank. Whenever possible, the seawater should be drawn directly from a tube well. Water from the tube well is usually clear and clean water is needed. It can be pumped directly into the filter tank. Then marine pumps. Centrifugal and submersible pumps are commonly used in hatchery operation. Centrifugal pump is more desirable in the big hatchery because it has a higher total head capacity. The selection of the size and type of marine pump depends on the size of hatchery and the daily water requirement. For the smaller backyard hatchery, a submersible pump with a discharge pipe diameter between 1 inch to 4 inch and a discharge capacity of 6 to 20 tons per liter is sufficient to provide the water needed. Medium and large hatcheries normally use centrifugal pumps. The size of the pump depends on the total water requirement per day and the maximum pumping time. Then the layout and the construction. Once the project site is selected and production target defined as an aerial survey of the proposed site, it will help to determine the perimeter of the area. An aerial view would show the important topographic characteristics such as rivers, shoreline, mountain, and low-lying plains. A master plan of the hatchery is then made. Layout of the hatchery should provide a schematic design of the location and integration of various facilities such as buildings, broodstock tanks, larval rearing tanks, nursery tanks, spawning tanks, pump house, air supply, and powerhouse. Laboratory, staff house, piping for water supply, and drainage canal. The layout plan should include the exact dimension, locality, shape, and size of said facility. Terminal drawing should show the detailed structure of the facility and size and quantity of support materials needed to ensure stability of the facility. From the technical drawing, the owner can roughly estimate the cost of construction. Since the technical drawing presents details of the structures such as thickness of the tank wall, amount and size of iron bars, and mixing proportion of concrete, the price of these materials will be easy to canvas to arrive at the cost estimates. However, estimations of construction cost should be done item by item before making the total estimation. In the total estimates, labor cost should be included. Life cycle. Based on their mode of life, pineid shrimps can be grouped into wandering and burrowing groups. Wandering groups means species belonging to this group have pronounced age groups from dense schools and are always on the move and active day and night. They prefer turbid waters with soft mud bottom, burrowing slightly or not at all into the substrates. A typical wandering species, Penis orientalis in the Yellow Sea migrates southward for over wintering and northward in spring to its spawning ground. The distance between the areas for overwintering and spawning is approximately 700 to 1300 kilometers. Other species belonging to this group are Penis setiferus of the Atlantic coast, Penis marginensis, and Penis indicus of the Southeast Asian region. These tropical species, however, do not need overwintering. Penis marginensis and Penis indicus generally spawn at a depth of 7 to 30 meters in offshore waters, usually near the nursery ground. The larvae spend part of their life cycle in nursery ground until adolescent stage. When they start migrating to deeper and more saline waters, since Penis marginensis, Penis indicus, and other wandering type of pineid shrimps from dense schools, fishermen can easily collect large number of spawners during peak seasons. Then burrowing groups. This group prefers certain habitats with sandy bottom. They do not have pronounced age groups. They show marked nocturnal activity, burrowing into the bottom substratum during the day and emerging at night to search for food. This diurnal activity is closely associated with changes in light intensity, but may also be due to other factors. Examples of this group are Peneus japonicus in the Japanese coast, Peneus guiarum of Florida, and Peneus monodon in Southeast Asia. Peneus monodon spawners are found in offshore waters on sandy bottom at depths of 20 to 40 meters. The larvae move towards the coast, enter estuaries and mangrove swamps, which serve as nursery ground. They then migrate to deeper water when they become adolescent and finally move to spawning grounds upon becoming adults. Peanut shrimps generally spawn in offshore waters. Each gravid female lays as many as half to approximately three-quarters of a million eggs per spawning. After hatching, the early larval stages like Nautilus, Protozoa, Mysis, and early post-larvae remain planktonic for some time and are carried towards the shore by tidal currents. The first larval stage is known as Nautilus. The unsegmented body, which is pyriform in shape, possesses three pairs of appendages. The Nauplius undergoes six moltings within 50 hours into a protozoa. The body becomes elongated with a distinct cephalothorax. The early protozoan stage has a pair of protruded compound eyes. The next stage is characterized by the presence of a rostrum, and the late protozoa stage has a pair of uropods. After 4 to 6 days, the protozoa finally metamorphoses into a mysis. At this stage, the larvae assumes the form of a juvenile shrimp, at which the pleopods are to develop. At this stage, tiny protrusion known as pleobasis are seen on the ventral side of the abdominal segments. The next stage is marked by the development of first segment of the pleopods development. The mysis remain drifting in the water column until they metamorphose into post larvae within 10 to 12 days. At post larval stages, the pleopods become fully developed and functional. The animals grow very fast in terms of size and are able to swim freely, although early post larvae are still planktonic in offshore waters. At a body size of 0.8 to 1 centimeter in body length, they enter estuaries and inshore shore waters, where they first adopt a benthic existence. The shrimps spend their juvenile, adolescent, and sub-adult stages in estuarine waters and then gradually move towards deeper water as they grow and eventually returning to offshore water when they attain sexual maturity. Inadequate supply of spawners remains as one of the major constraints in the development of the shrimp farming industry, especially for species such as the tiger shrimp, Penis monodon. Recent success in eye stalk ablation techniques has greatly enhanced constant supply of spawners through ablating captive broodstock to maturation. Now, what are the conditions of broodstock? Upon arrival in the hatchery, the broodstock are first acclimatized in holding tanks for 4 to 7 days. The holding tank should be big enough to provide proper space and aeration. Then 60% of the water in the tanks is changed daily. Once the animals have recovered from transport stress, they are induced to molt by manipulating salinity of water in the holding tank. The salinity is decreased by about 4 to 5 ppt for 2 days and then increase to the normal salinity of the seawater. Majority of shrimps will molt after changing salinity. Mating occurs during this time when the females are newly molted. The shrimps are then ablated 2 to 3 days after molting or when the shell is completely hardened. Then induced maturation. Only suitable broodstocks are used for eye stalk ablation. The criteria are complete appendages, presence of spermatophore in the thylicum of females, size should at least be 100 grams. Presently, the most practical way of inducing maturation is by unilateral ablation of either right or left eye stalk of the female. Ablation is done by using a razor blade to cut or open the eye, then squeezing out the eye stalk from the base to the tip with the thumb and forefinger or using the fingers alone to break and squeeze the eye. The ablated animals are stocked in maturation tanks at a density of 5 to 6 per square meter and a sex ratio of 1 male to 1 female. Then the broodstocks are fed with squid, mussel, or cockle meat or pellet feeds at the rate of 10% of total biomass. The water in the tank is allowed to flow through continuously or changed daily at 6 to 70% of total capacity. Then sampling. Gonadal development of an ablated female is checked 3 to 5 days after ablation, while checking for gravid females is carried out every other day. Sampling and checking are done at night or at any time if the tanks are sufficiently covered and kept dark. During sampling, an underwater flashlight tied to a pole is held close to the shrimp so that the light strikes perpendicularly on the dorsal part of the body, where the ovaries are located. Water in the maturation tank can be lowered to 30 centimeter to allow the worker to get inside for closure observation. Only gravid females with stages 3 or 4 ovaries are collected and transferred to spawning tanks. Coming to larval feed, under natural conditions, pinead shrimps are either omnivorous scavengers or detritus feeders. In general, shrimp larvae feed on phytoplankton, detritus, polychaetae, and small crustaceans, and their food preference changes with age. They start feeding at protozoa stage. The protozoa and early mysis stages prefer phytoplankton, though the digestive system is not yet fully developed. At mysis and early post larvae, food preference changes to zooplankton such as rotifers or brine shrimp. As the larvae grow older than P6, feeding habit changes again to that of a bottom feeder. Polychaetes, chopped mussels or cockle meat are fed during these stages. Large-scale production of phytoplankton for larval rearing can be obtained in two ways: by direct fertilization of seawater in the rearing tanks or from pure culture. Many workers in the field rely partly on mixed populations of the phytoplankton present in rearing water. In the past, fertilization of tank water was done directly with the resultant algal population from the blooms utilized for feeding purposes. The method, however, has its advantages. Using direct fertilization techniques, heavy blooms of undesirable species often occur. Such circumstances have led to the development of techniques for screening different algal species suitable for larval culture and isolation techniques for pure culture of specific types of phytoplanktons. In addition to this, many other types of feed have been developed and tested as part of perennial shrimp larval rearing strategies. These feed may either be frozen or dry material from mollusks. Crustacean tissue, soybean cake, soybean curd, egg mustard, and fertilized eggs or oyster. Other types are available as formulated diet or the newly developed microparticulate and microencapsulated diets. However, the choice of a particular feed used should be properly evaluated based on the following criteria: availability and ease of handling, then coming to feed performance and feed cost and rate of return on capital. Phytoplankton culture. The more popular phytoplankton species found to be suitable as food for the early larval stages of shrimp are Ketocirrus calcitrans, Skeletonema costatum, Tetracelliums species. These plant components of the oceans usually require certain environmental conditions for growth. In the wild, phytoplankton abundance is easily affected by fluctuations in temperatures, day length, presence of grazers, availability of nutrients, water depth, and turbidity, as well as the seasons of the year. Manipulations, however, of the above mentioned factors is possible under laboratory conditions or controlled rooms. Phytoplankton culture is usually carried out by subjecting a known amount of phytoplankton to an environment suitable for its growth. Next, how to use rotifer culture. The rotifer Brachiones plictalis, Brachiones plicatilis is the most important zooplankton utilized as live food for various cultivable marine animals. Culture of rotifers usually involves the use of various algae like chlorella and tetraselmis, as well as other food items like bakers and marine yeast. Brine shrimps as larval feed. Many hatcheries depend largely on brine shrimps to feed their growing shrimp and fish larvae. In most cases, it is the nopli that are used to feed the larvae, probably because of its small size and relatively slow moving habits. They can be easily preyed upon by the predatory fish or shrimp larvae. Preparation of egg custard, contrary to the use of boiled egg yolk. Preparation and use of egg custard is preferred for use as larval feed. Chicken egg is broken open and the egg white and yolk placed in a bowl and a small volume of water while manually stirring the solution or mixing can be done with the use of an electric blender. Then steam the homogenized solution until it hardens. The egg custard is then passed through a sieve with a desired mesh size. Weigh the particulate egg custard according to the desired quantity, that is mg for number of larvae. Then, contrary to the use of boiled egg yolk, preparation and use of egg custard is preferred for use as larval feed. Chicken egg is open. Then, selection of spawners and egg collection. Although spawning occurs throughout the year among tropical species of shrimps, there are distinct periods of the year when majority of the shrimps spawn. In the case of Peneus monodon and Southeast Asian waters, there are two pronounced spawning seasons from December to March and June to September, with only one pronounced period. Example, June to September in the case of Peneus merginensis and Peneus indicus. While spawners of these species of shrimps are supposedly available all year round, they are abundantly caught during the spawning season. A gravid female can be easily recognized by the presence of a very large and distinct diamond-shaped dark green mass of ovary between the first and second abdominal segment below the dorsal shell. Selection of spawners can be easily done by holding the animal with its ventral body facing a light source. The criteria used for selecting spawners from the wild are stage 4 ovary, complete appendages, then the back is not broken, presence of spermatophore underneath the thylacium, the color of the shrimp, especially Penis monodon, should be pink with a faint greenish tint. Spawners which are slightly reddish could be due to stress caused by abrupt lowering of the water temperature during transportation by fishermen who try to delay spawning. Stressed spawners give very low spawning rates. Procurement and transportation of spawners. The major fishing gears in collecting spawners can be broadly grouped into three categories: stationary trap, mobile, and entangling nets. Then after that, we should go for treatment of spawners. Then we also need to check into spawning activity. Then we need to collect the eggs and then treat them. Then after all this, after spawning, the animal is removed from the tank the following morning. The eggs are then cleaned either by siphoning into egg collectors or draining 2 by 3 of the water through a filter net that effectively retains the eggs within the tank. When draining is completed, the scum is then removed using a scoop net with a mesh size bigger than that of the shrimp eggs. The tank is then filled up with new seawater. During the cold season, fungus and bacteria are likely to infect the eggs during incubation. Preventive treatment normally consists of dipping the eggs in 1 ppm of methylene blue or 0.5 ppm of malachite green for 10 minutes or 3 ppm KMNO4 for 30 minutes. After that, the eggs are transferred to a cleaner tank for further incubation and subsequent hatching. From the incubation of the hatching tank, samples of eggs are counted to determine the number of eggs spawned per female. Then hatching and transportation of nopli. Eggs of most species shrimps within 12 to 18 hours after fertilization at temperature and salinity range of 26 to 30 degrees centigrade and 30 to 23 ppt respectively. The density of nopli is estimated a day after hatching. Nopli from 300 ml water samples taken from the spawning tank are counted and averaged. The total number of nopli in the tank is then obtained by multiplying the volume by the average density. Then how to transport the nopli? By using plastic containers. Only strong and healthy larvae should be transported. This is done by concentrating the nopli at the water surface with a light source at night. Gather them by scooping with a plastic or glass container. The larvae are then transferred into plastic jars which are half filled with seawater. The container is then gradually filled up. A 20-liter plastic containers can contain a maximum number of 5 lakh nopli. The open end of the container must be properly sealed to prevent leakage. The survival rate after 6 to 8 hours during transportation is more than 50%. Plastic bags. Each bag containing about 6 to 8 liters of water can be stocked with 2 lakh nopli. The water in the bags is oxygenated and the open end is closed with the rubber bands. The survival rate is about 80 to 90% if transportation takes about 4 to 6 hours. After that, routine hatchery management, which includes the maintenance of optimal environmental conditions, that is to necessitate the maximum growth and survival of the cultured organisms. Then we need to check into maintenance of water quality. We have to check the salinity, temperature, dissolved oxygen, pH, and nitrogenous compounds also. Then, with this, we have come to the end of this chapter. From this chapter, these are the type of questions that we might get. Answer the following in about 30 lines. Write about selection of a suitable site for shrimp hatchery. Write about shrimp hatchery design and construction. Write about hatchery facilities. Write about seawater supply and piping system. Write about life cycle of wandering group. Write about life cycle of burrowing groups. Write about preparation of brood stock for spawning. Write about shrimp larval feed. Write about procurement and transportation of spawners. Write about hatching and transportation of nopli. Write about shrimp larval rearing. Write about shrimp larval rearing in small indoor tanks. Write about shrimp larval rearing in large nursing tanks. Then write about maintenance of water quality in shrimp hatchery. Write about shrimp nursery of post larvae. Write about harvest and transport of shrimp larvae. Hope you all have understood. Thank you.